Yes.
Okay, so hi everyone, and thanks Dennis for organizing everything. So I figured I'll just have an opportunity to introduce everyone to the genetics of amyotrophic lateral sclerosis, or ALS, and frontotemporal dementia. So you may have heard about ALS through the famous ALS Ice Bucket Challenge. So this is the man, Pete Frates, who started the ALS Ice Bucket Challenge outside of Boston College here in Massachusetts. So he did develop ALS at only age 27. And he really caused a wave of explosion. It went all the way, really all around the world. And people would pour water over their head in support of this relatively unfunded but very important disease. And this collected so much money and was able to help ALS research throughout the world. This is actually my lab from Canada back home, us participating in the ALS Ice Bucket Challenge. So it was great to definitely participate and raise awareness as well as funds for this disease. So amyotrophic lateral sclerosis, or ALS, is characterized by degeneration of motor neurons, leading to progressive weakening of limb, bulbar, and respiratory muscles. So we have these motor neurons in our nervous system. So we have upper ones as well as lower ones down here. And they control voluntary movement. So they assist in doing really any daily tasks. But as the disease progresses, it starts to affect involuntary movements as well. And this often leads to respiratory failure. Currently, there's no cure for the disease. There's only one FDA-approved drug, right here, riluzole, and it really only extends life by only 3 months. So this is not a huge improvement, and definitely needs more attention. And by doing a lot of genetic research, hopefully we can identify more druggable targets. The disease is so important and so really so fatal is that it actually ended up being the main disease, one of the main diseases that overturned the Supreme Court of Canada ruling in favor of legalizing doctor-assisted suicide. Because again, the death due to respiratory muscles can be so severe, and because there's no treatments available, this is why we now have doctor-assisted suicide in Canada. Up to 10% of cases are considered to be familial because they do have a family history, and the remaining is sporadic. However, as we do more studies, we are seeing less of a difference between sporadic and familial cases. In the clinic, they're not really distinguishable. So it's just, do you have a family history, and can that ever be genetically tested? There are some forms of ALS where dementia can occur, or Parkinson's disease. So this is another disorder, and for a long time it was actually considered completely separate to ALS. So this is called frontotemporal dementia. So this is atrophy of the
There's also definitive hematopoiesis, and that the normal macrophages, originate from a different lineage, not from this one.
So it's just background. Someone unmuted themselves.
Okay. So again, we have atrophy of the prefrontal and anterior temporal cortices. There's 3 main types of frontotemporal dementia, or FTD. There's behavioral variant FTD, primary non-fluent aphasia, or semantic dementia. So there is comorbidity with ALS in 15% of FTD cases, and up to 50% of ALS patients have some signs of cognitive impairment. So what causes this disease? Let's focus on ALS for today. There have been reports of environmental influences, heavy metal poisoning, so this is due to lead, mercury, selenium, or zinc. Smoking and pesticide exposure have also been linked to the disease. There's also strenuous physical activity at a young age, so there are many reports describing athletes that are more susceptible to ALS than the general population. So this is typically athletes that participate in sports where concussions are a lot more frequent, causing repeated damage to the brain, and over time this develops into ALS. So football, hockey, those kind of sports. There's also been a very interesting association with this amino acid from cyanobacteria, so beta-N methylamino L-alanine, or BMAA for short. So I'll talk a little bit about this in the next slide. So in 1945, there was a report that the incidence of ALS in Guam, which is a Pacific island that belongs to the United States, the incidence of ALS was significantly higher compared to the rest of the world. So in addition to ALS, there is Parkinson's disease and dementia within this subpopulation, the Comoros population of Guam. And it was later discovered that this BMMA is the neurotoxin was identified in seeds from a plant that were used by the locals to make flour. So many of them were constantly ingesting this neurotoxin unknowingly, and this led to a much higher incidence of ALS in Guam. So this was also validated by postmortem analysis, which revealed toxic levels of this neurotoxin in brains of ALS patients from Guam compared to healthy ALS-free controls. However, further investigation into Guam as genetic sequencing or genetic studies really became more commonplace, they did actually identify that some of the patients who died from ALS did have an elevated genetic risk for ALS. So perhaps it was both this exposure to this neurotoxin plus the genetic background that led to a higher incidence of ALS in Guam. So that was really an interesting case because it demonstrates that ALS can be caused by environmental factors. So let's look at some of the other factors that may contribute to ALS. So genetic factors, there have been Mendelian forms of ALS. The most common is autosomal dominant. Rare forms tend to be autosomal recessive or X-linked. There are also complex susceptibility genes underlying ALS. So many of these genes have been implicated in disease susceptibility and identified through genome-wide association studies, where the former have been identified in linkage analysis. The problem, though, is that ALS is such a progressive disease that a lot of times many individuals don't survive to be included in any linkage analysis studies. So this is one table from a great review that was out in 2014, just demonstrating some of the more common ALS genes to be mutated. So here they are located here with their respective inheritance, the percentage of familial ALS that they explain versus sporadic. And I'll talk a little bit about why these numbers can differ based on bias ascertainment and, of course, their putative function. We'll talk a little bit about the pathways that these genes are involved in later on, but the idea of ALS linked to autophagy dysfunction or RNA metabolism is a recurring theme in this disease. There's also other genes that are implicated in the pathogenesis of ALS, but appear less in cases. So these are listed here. Whether they fully cause the disease or just give a heightened risk is yet to be determined. With the benefit of next generation sequencing being more affordable and available, we could definitely answer those questions. We could also look outside of just familial ALS and consider whether more sporadic ALS cases have also been observed to have mutations in these genes as well. And again, here's their putative protein function. So putative because these genes are still under consideration. So looking at the timeline, there's about 15 to 20 genes associated with ALS, and this is Mendelian as well as risk. And it wasn't actually until 1992 that about 13 missense mutations were identified in a gene called SOD1. So this was the first gene to be identified in ALS. So here on the y-axis on the top half explains the proportion of familial ALS cases that can be explained by mutations in these genes. In contrast, this is the proportion of sporadic cases that can be explained by these genes. And again, I'll talk a little bit about why this graph can be misleading in some ways. So, so again, in 1992, we had the first gene discovery, and then really we just keep growing, and there's been many more that I'll talk about later on because of the benefit of next generation sequencing. So Next, there was the TDP43 gene, the FUS gene, and later on we discovered the C9orf72 gene, which really explains the majority of ALS cases to date, both familial and sporadic. So again, breaking it down again, these genes, you could see we have the genes discovered to date explain about 70% of familial cases, where 30% is yet to be elucidated. In contrast, sporadic cases, the majority of them are still unexplained. So the caveat, though, here is I've kind of alluded to earlier, is that we do treat FALS, so familial ALS, very differently than sporadic cases. A lot of the times the clinician would ask, is there a family history? And there are more individuals available. However, in sporadic cases, if a single patient walks into the doctor's office and there's no family history, it's automatically labeled sporadic. And a lot of the times they are genotyped for variants that were previously identified in familial ALS to see if that's what's causing it. So as you can see, this is not really the approach that we want to take today. So you'd want to sequence these individuals for the entire FUS gene, for example, to truly say that they are, they don't have a FUS mutation, rather than just genotype individuals for a FUS mutation that's been previously associated, because perhaps that FUS mutation was unique to that family or was common to a certain isolated population. But of course, this is very expensive. Even today, it's still a little bit impractical for the average clinician. So as sequencing goes down in price, clinicians can then see the opportunities to test their sporadic cases for all genes that are involved in ALS, and not just genotype them, but really sequence them fully. And I would anticipate that this number would actually grow significantly, and these bars would really reflect a little bit more of what we see in familial ALS cases. And so this review was published in 2014, but even again, a paper that I'll show later on in 2015 is already showing this number to be a little bit bigger. So you can see the benefit of sequencing is already working. So SOD1, again, was the first gene to be discovered in ALS. And to date, there's over 100 mutations identified in SOD1. And again, it explains about 12% of familial ALS and 1% of sporadic cases. There is one variant, interestingly enough, alanine to valine at position 4 in the SOD1 gene, and it's the most frequent variant in North America. And what's really interesting about it is that it's just a missense mutation, but it gives rise to a very aggressive form of ALS. Patients who have this specific variant often die within only 1 year. So this is actually quite a significant mutation. In contrast, in the same gene, individuals with this specific mutation and aspartic acid to alanine change at position 90, even in the homozygous state, can actually live much longer than the previous mutation, and up to 10 years. So this is a very interesting question that many people are still trying to wrap their heads around. Why can this happen in a single gene? There are many clinical trials that focus on SOD1 because it was very exciting. It was the first gene discovered, but there is a big problem of only honing in on SOD1. First, again, it explains a small proportion of ALS, and as we found out later on, it actually lacks the classic pathology presentation of ALS. Typically, we see inclusions of a gene of a protein called TDP43, which I'll describe in the next slide. We don't see this in ALS cases with SOD1 mutations. So perhaps focusing all the efforts should be redirected to the other genes as well. So this is the gene here. This was a major significant finding for the ALS field. TDP43, encoded by the gene TARDBP, is a major component of the ubiquitin positive neuronal inclusions, and these are the pathological hallmarks of both ALS and FTD. The reason that this was so significant was because it also started to link these ALS and FTD, which were for a long time considered separate clinical entities. So to link them together may explain why some individuals with ALS also develop cognitive impairment and eventually frontotemporal dementia. And then vice versa, why some individuals start off having frontotemporal dementia symptoms and then they develop motor neuron disease later on. And later on, it was also discovered that specific mutations in this gene are observed in both ALS and FTD families with an autosomal dominant inheritance. Interestingly as well, the majority of the mutations linked to ALS tend to be at the C-terminus of this gene, and this is specific to RNA processing. So this is, again, a repeated theme in ALS. And right now, it accounts for 44% of familial ALS cases, but the actual observation of these inclusions occurs in the majority of ALS cases. There's another mutation, FUS, and it explains up to 4% of familial cases. It was originally linked to autosomal recessive, but now dominant ALS as well. And the reason this was such an exciting finding was it really confirmed what we were seeing with TDP-43. It shared a lot of the functional homology, so mutations that were observed in FUS tend to cluster around the C-terminus of the gene, and this is where the RNA binding protein, RNA binding domain is located within the FUS protein. Probably one of the most exciting findings for ALS was this gene here, C9orf72. The two papers that discovered these genes were back in 2011, and by the NIH as well as the Mayo Clinic. And this was just a huge discovery for ALS, really well-cited papers, well-cited, or really nice research coming out of both groups. So this gene here, as you know, is still a John Doe, so chromosome 9, open reading frame 72. And the reason it's still called as such is because we still don't know what it's doing. But there is an expansion in the intronic version, depends on which transcript you're examining, of this gene. So it's 4 glycines followed by 2 cytosines in a row, and many individuals who present with ALS and FTD often harbor this hexanucleotide expansion in this gene. So again, linking the two diseases again and implicating that they're probably a continuum rather than separate clinical entities. So much similar to the Huntington's expansion we see, the repeat size can range and the severity can also range. So it can range from 2 to 20, which is typically common in the healthy population and likely benign, 2 to a few hundreds. And this is kind of this uncertain risk. You may have a heightened risk plus perhaps environmental exposure, or is this sufficient to cause the disease? And then the last category, which is pathogenic, and this is when you have over a few hundreds to thousands repeats of this specific, in this specific gene. So what is exactly this gene doing? It's been really it's starting to be well studied, but we don't fully know what it's doing. There's a few different hypotheses. So the first is that haploinsufficiency leading to neuronal degradation through the endocytosis and pathway and infecting autophagy as well. There's the idea that sense and antisense transcript accumulate in nuclear RNA aggregates, and this then goes on to recruit RNA binding proteins. And as I mentioned, this is a recurring theme in ALS pathology, so this idea was quite appealing. Finally, the gene can also undergo repeat associated non-ATG translation. So this is an unconventional form of translation leading to expression of 5 specific dipeptide repeats containing proteins, and this ultimately goes on to form the inclusions in the brain that we see in patients with ALS postmortem analysis. So this gene, there's still a lot of activity happening on it, but because of these different hypotheses, the application of antisense oligonucleotides may be the appropriate therapy to at least cater to the proportion of patients with ALS and FTD who carry this expansion. There's also other interesting forms of ALS. So this idea of de novo variants in ALS, there have been observed before in a sporadic ALS case where a sporadic SOD1 mutation was observed, and also in SALS trios with sporadic FUS mutations. The question that we really want to ask is that, do they really cause the phenotype? So the presence of mutations in these ALS genes in cases and absent in siblings and parents may suggest that the de novo variant is causing the disease, but I think it's really good to functionally validate these mutations and provide more evidence to see if these de novo variants are indeed causing the disease. A huge limitation, of course, of doing or testing de novo variants is that the age of onset of ALS is much later than, say, the age of onset for autism, therefore preventing access to parental DNA and the proband. And so to do these trio analyses would be quite difficult if the individual develops ALS, say, in their 60s, which is quite common, and their parents are deceased. You may overcome this by having a sufficient sample size of siblings to see what the parent's genotype would have been as well, and determine if it is truly de novo. There's also common variants in risk loci in ALS. So there's up to 14 studies that conducted genome-wide association studies on ALS, identifying various genes, some that I've highlighted before and some are a little bit new here and have not been replicated for the most part, but many of them have been. And they also show their effect size in this paper as well. So typically modest effect sizes of these risk loci. But the nice part about this specific paper is that it used genome-wide association data from 3 studies to estimate the heritability of ALS. So you could see, previous to this paper, we thought the heritability of ALS was actually much lower, but now it's up to 21%. But this is including all subtypes, where if you look at based on group, based on how the disease actually starts, you could see that some of them look a little bit more heritable. So this is something that's really interesting, but then it just also goes to show us that we're still needing to do much more genetic research to identify the rest of the genetic determinants of this disease. Another great study looked at the burden of rare variants in ALS. So this idea that in addition to having a single variant, perhaps you have multiple variants. So you could see these are the patient's type here, so sporadic with some familial, and the first variant they identified versus the second variant that they identified, and the possible model. So many of them were homozygous, some of them were autosomal dominant, but this really neat idea that keeps coming up in ALS research and still needs to be validated is this idea of oligogenic inheritance. So the idea that you have 2 or more rare variants that are occurring in a gene that may be modulating the phenotype. So perhaps the expansion that we saw in this gene is causing the majority of the phenotype, or can explain the majority of it, but this additional variant is modifying the phenotype, creating perhaps the dementia part, or leading to a much earlier age of onset or a worse disease outcome. So how they're modifying the phenotype really remains to be elucidated, but this is really neat that we're observing multiple mutations in a single case. And this would have really not been possible had next generation sequencing not been available. So it's great that we're already seeing it. So as I alluded to earlier, the way we treat sporadic cases from familial cases is problematic, and we don't usually test sporadic cases the same way we test familial cases. Again, we tend to genotype them rather than fully sequence them, but using the same analysis of sequencing here for both sporadic and cases, they also saw that the percentage of subjects with variants in ALS genes is much higher than initially anticipated. Yes, still lower than familial cases, but still much higher. There's also been novel gene discoveries in ALS, which is, again, very exciting thanks to next generation sequencing. One of the most, the biggest paper that came out in 2015 was exome sequencing in ALS, identifies risk genes and pathways. This led to the great discovery of TBK1, which is a gene that was implicated in known ALS pathways, but the gene itself had not been linked to the disease until this study here. Another gene, NEC1, was very exciting to initially be reported by Cerulli et al. And then this summer as well, we saw a lot of excitement by a group at the University of Massachusetts confirming the association of NEC1 in ALS. So this was a great finding, and what's really actually exciting is that the majority of the funds to conduct this research, which led to NEC1, was funded by the ALS Ice Bucket Challenge. So that was wonderful. We also see the usual suspects appearing here again, giving credit to the study, meaning that they had done their methods well. SOD1 again, and TDP43, and this other gene, optineurin. So part of optineurin actually binds to TBK1. They're linked in the same pathway. So this again gives further evidence to the role of TBK1 in ALS. Whether it's fully causative alone, like if it's Mendelian, or if it's a risk allele, remains to be identified. Some groups have described TBK1 haploinsufficiency to be a cause of ALS, but I think more research is needed to really confirm how TBK1 causes ALS, what the exact mechanism is. And this is again just a QQ plot showing the typical genes that are associated with ALS. So here again, SOD1, our favorite gene, still very much associated as we would expect, TDP43, and right here is TBK1 as well. And then I don't think NEC1 is displayed here. So which pathways are affected? So again, as I described, the novel gene discoveries were very exciting, but they also confirmed the known ALS pathways. So this was great. It means that we have the certain pathways targeted and can focus on those. And as you can see here, there's multiple genes with multiple cellular roles. So we can see that TBK1 is involved in autophagy here, and then the NEC1 is involved in DNA and RNA processing as well. But you could see here's the C9orf72 gene involved in potentially autophagy as well as RNA processing. So which pathway is involved? Some of them are unique to a certain pathway, and then some of them are again involved in these pathways that are recurrently disrupted in ALS. So some goals and priorities in ALS research that I think we should focus on moving forward is to elucidate the function of these ALS genes. So after discovering them in genome-wide association studies or burden analyses, we should really look into the full function of these genes. What do they do? A lot of studies focus on developing mouse models of a single gene. This may or may not be the best approach. As I've illustrated before, it looks like multiple genes are implicated in even a single case. So perhaps we need to be a little bit more liberal in how we think about functionally validating these descriptions. It's important to also characterize these pathways fully. Again, as I described, the autophagy pathway and RNA processing pathways have always been recurrently mutated or disrupted in ALS. So focusing on these pathways and the fact that multiple genes are involved in these pathways is great, because then you could target the pathway rather than a specific gene, catering to a much larger population of ALS patients. I think it's really important to conduct large-scale genetic analyses to identify novel genetic factors as well as confirm the ones that are currently described in the disease. And we hope to do that here at the Broad as part of my postdoc. We'll see how things go. It's also really important to look at conducting genetic studies, but for non-European cohorts, the majority of the genetic analyses to date have focused on European cohorts, but it's definitely really important to go into the Asian, the African population as well. And finally, and most importantly, translate our findings into clinically relevant targets, as this disease is, again, currently incurable. So definitely need to focus on how our genetic findings can help these patients. Finally, if you have any questions, here's my email. It's sfarhan@rhodeinstitute.org. And I'd also like to thank the ALS Canada for providing me with postdoctoral funding through the Tim E. Knoll Postdoctoral Fellowship. So thank you very much.